Alrighty, good morning. Going to talk a little bit, sorry, wrong one. We're going to talk a little bit about genetics this morning. We're going to talk about somebody that was very important to the field of genetics. Sometimes it's called the father of genetics, and that would be Gregor Mendel. So, who Gregor Mendel was? He was actually an Austrian monk, and in his, if you could call it spare time, he studied statistics and a little bit about science, some botany. He was very, very interested in plants. He carried that interest into tending for the monastery garden. And when he was doing that, he noticed some he just had some things that he noticed about some of the plants and wondered how certain traits got passed from parents to offspring. And so one of the plant that he is most well known for working with is the pea plant. And so as he was noticing these different traits that the different plants had, he kind of just wondered, how did those things get passed on? And so he decided to work with pea plants to see if he could develop any sort of patterns, or if he could figure out how traits got passed on from a parent to an offspring. What I have there is right here is the science. Well, there, my mouse went away. Right. Well, okay. So my mouse is not going anywhere near it, but it's right here, the Pisum sativum. That is the Latin name or the binomial name for a pea plant. Kind of a side note, but with scientific names, there are always two names. The first name you capitalize, the second name you lowercase. It's also called the genus species. So the genus for the pea plant is Pisum. And then the species, a little more specific version of that genus, is Sativum. That goes into the classifications, the whole big system of kingdom, phylum, class, order, family, genus, species. That's just how we categorize and name all living things. So genus, species. And like I said earlier, he is kind of considered to be the father of genetics because he was the first person to really look and study the patterns of inheritance in any living organism. So there's a nice picture up there in the right corner of him. So nice portrait. So kind of going to go into his pea plants and why he chose the pea plants to work with. They're actually a very good organism to work with for three reasons. The first, they had many different traits. You could study their stem length, you could study their flower color, their flower positioning, you could study the seed color or the seed shape. If I remember, there's something like 8 or 10 different characteristics, and for a plant, that's quite a bit. And the other bonus with these numerous traits that he could follow is they only had 2 different forms. So it was either one thing or it was going to be another. There was no middle ground. So like with humans, for example, we have any number of different heights that people can end up being. There's no 2 heights, that people are either this height or that height. But with pea plants, their stems are either tall or short. There's no medium stem length in pea plants. You're either tall or you're short. Your seeds are green or they are yellow. There's no other colors to pick from. The seeds are wrinkled or the seeds are smooth. There's nothing in between. You can either have white or purple flowers. So it made it really simple and really easy to track the traits. So you could take a plant that had white flowers and cross it with a plant that had purple flowers, and you could see which trait would overtake the other and appear in the next generation. So the second reason why pea plants were such a beneficial organism to use in studying genetics is they produce large amounts of offspring per generation, which allows you to have numerous test subjects to look at per generation. And since they are so quick to reproduce, you can watch the inheritance patterns over many generations. It's difficult with humans because our average lifespan is something like 80 years. So there's not really a good way to follow and track genes through generations on humans, because you probably are only going to be able to witness 2, maybe 3 generations, depending on how far spaced the family is. So, but with pea plants, you could literally have hundreds and hundreds of generations, within 1 year that you could follow and see how those traits were passed along. And the third reason why pea plants were so beneficial is it was very simple to determine what traits they wanted to cross. It was very easy to control that. We call this selective breeding. When you take a certain trait and you specifically breed that trait, or you take this trait and you breed it specifically with another one. Think of dog shows and horse racing or horse showing. They specifically try to breed certain traits into or out of the litter. So for this, it was very easy to say, okay, I want to see what happens when I cross this tall plant with this shorter plant. So it's very easy to manipulate that. So the way he did this is to be able to track the traits. He allowed the pea plants to self-pollinate for a couple of generations. He wanted to make sure that he had, quote, pure traits or pure plants. So, meaning they didn't have, we'll take stem length. If the tall stem was dominant and would always cover up the short stem trait, he wanted to make sure that all of the plants that were tall carried both tall genes. He didn't want the plants to have one tall and one short. And it was really difficult to tell which ones were tall and which ones had both genes for the tall, and which ones had a tall gene and a short gene, because they look the same. The short gene just gets covered up if you happen to have it. So he let them self-pollinate, and any plant that came out with that less common, or what we call a recessive trait, would just not be allowed to reproduce. So he tried to get those pure traits of plants that were either all tall and carried both tall genes, and then all of the plants that carried the short genes. So when he had those pure traits, he called this generation the pea generation, or the parent generation. Okay, so first thing he did to study is try to create that pure generation, or that pea generation, the parent generation. So that now he's going to take those pea generation plants, and he's then going to start cross-pollinating. Cross-pollination means that you are taking the pollen from one plant and fertilizing the seeds of another plant. Self-pollination, it sounds like what it is. You are taking the pollen of one plant and fertilizing the seeds of that same plant. So it's self-pollinating. But he, in this case, is cross-pollinating. He's taking pollen from one plant, crossing it with the seeds of a different plant. So you're getting a mix of genes at this point. So I took seed color in this instance. So again, since there are only two different versions of the traits, it was easy to do that. So he took a pea generation that had yellow seeds and a pea generation that had green seeds, and then he cross-pollinated those. The offspring of this cross was called the F1, or the first filial generation. When you do genetics problems, people will ask you what were the genotypes of the P generation, or what were the phenotypes of the F1 generation. So you want to make sure that you know what the P generation and the F1 generation are. So we've got basically the kids of the parents. That's the F1 generation. He then would take specific plants from the F1 generation and cross-pollinate those to create a third generation, which was called the F2 generation. So these would be like the grandkids of the P generation. Now, he could go further on, but usually in genetics, we don't go past the F2 generation. And again, the F stands for filial, so it's the second filial generation. So most genetics problems are really only going to have you work with the P generation, the F1, and the F2 generation. So, like I said, just make sure you know what those two are. And from this, he was able to determine what traits were dominant, meaning they showed up if they were present, and which ones were recessive, meaning which ones got covered up if they were there. With this, he was also able to come up with two laws. The first law he came up with, or he was able to devise, was called the law of, that should be segregation. There we go. The law of segregation. I can't spell apparently, sorry. So this kind of is going back into meiosis that we talked about the other day. So what the law of segregation means is that the pair of alleles, so an allele is the version of the trait that you get from mom or the male, and then the other version of the trait that you get from the female or mom or dad. I can't remember. I'm sorry. You get one from mom and one from dad, or one from the male and female. So, and those two together are called alleles. Those are just different versions. So in the case of the pea plants, the trait might be seed color, and then the alleles would be the versions of that seed color, which would be a green trait or a yellow trait. And that version is just called an allele. So you could say a green allele or a yellow allele. So what the law of segregation is saying is when you are going through meiosis, or when you're forming the gametes or the sex cells, you separate those. You don't inherit both copies from mom or both copies from dad. You only receive one copy from mom and one copy from dad. But since mom has two copies from her parents, her body essentially, remember, decides which trait she's going to pass on. Is she going to pass on the trait she got from mom, or is she going to pass on the trait she got from dad, from her dad? So that ensures that you end up with two copies of every trait, or two alleles. So he was able to determine from his studies that those alleles have to be separated during meiosis. So the two parent plants, they've got, let's say they've got 7 different, for all intents and purposes, let's say there's 7 different characteristics in a pea plant, which means that a pea plant would have 14 chromosomes then, because the pea plant would have gotten 7 chromosomes from its male parent and 7 chromosomes from the female parent. But it's not going to pass on both. So during meiosis, that one parent is going to separate those alleles, so it only passes on the 7 instead of the 14 that it actually has. So, and this explains why we've got 4 haploid cells at the end of meiosis instead of 2. Because if we had 2, you'd have 2 copies of the allele in each cell. The second law he was able to come up with was the law of independent assortment, which just basically says that, for the most part, traits are going to separate independently from each other. So the green allele that codes for green seeds is not tied to the trait that says the seed will be wrinkled. So they separate independently, so they're all separate entities. Now, there are some traits in some organisms that are linked, but for the most part, each trait separates independently of one another during meiosis. So that's how you could end up with so many different combinations. If they were linked, let's say that seed color and seed shape were linked, then that means that, if you had green and wrinkled seeds, if you're going to get green and wrinkled seeds. You're not going to get green and potentially smooth seeds, because that's not there. That's not what that particular plant had. So this, like I said, this allows you to have so many different combinations in the offspring. All right, a couple of vocabulary words to kind of introduce you to. When we talk about genetics, all we're talking about is just a branch of biology, so living things. Specifically, in genetics, people study heredity, which is the passing on of traits from parents to offspring. And I've got in parentheses there, traits just means genes or the DNA. So literally passing on the DNA that they were given from their parents. So that's what genetics is. Now, obviously, we can break this into a whole bunch of different subcategories. With different organisms. I mean, you can study genetics in plants, people, animals. You can study, you can study a whole bunch of different stuff. A lot of times what people use are genetic counselors, and those people are there to track diseases or specific genes that people want traced to see what are the odds that this particular gene would get passed on to a future offspring. The genes, we've kind of been talking about those. Like I said, it's the DNA. It's the characteristic or the trait that parents pass on. So I've given a couple examples here, like your eye color is a gene, your face shape, your hair type, your hair texture, nose shape, things like that. So the allele, like I said, is the different version of that gene. So you're going to get two different eye color genes, but the different versions are alleles. Same thing with your face shape. So I've given you examples for each one. So you've got the blue-eyed gene or the brown-eyed gene, but those are called alleles collectively because they're different versions. So just different versions is what an allele is. Tomorrow you're going to want to be aware of these. We'll all review them on the notes, but just to kind of put these in your brain right now. Genotype versus phenotype. What a genotype is, this is the set of alleles that a person has, or a plant or an individual. Doesn't tell you what you're going to look like, it just tells you what genes you have, because that's what your phenotype is. That's what you are going to actually look like. That's what the genes will translate into. So your genotype is literally going to be denoted by two letters. I'll try really hard in the genetics problems to not put in letters that look similar, uppercase and lowercase, because that matters. The uppercase and lowercase does matter. So my example of S's, that would be a bad one normally. P's, I probably won't use. I'll use something like the letter B, because you can definitely tell the difference between an uppercase B and a lowercase b. Your phenotype is what do those genes then translate into in terms of what you're going to end up looking like. So it's a description. So if I were to ask you to identify a genotype versus a phenotype, any genotype is going to be a set of letters or set of alleles, and then you'll automatically know that it's a phenotype because it'll be a physical description. All right, last slide that we're going to go over today is the dominant versus recessive traits, which we kind of talked about earlier with Mendel. The dominant trait is the one that will cover up the recessive trait if it's present. So there are two ways you can show this. Number one, you could have both copies of that dominant trait. The second way is you might have one copy of the dominant trait and you might have one copy of the recessive trait. But because you have that dominant trait, you will automatically show that dominant trait. My example that I've given you is brown eyes. Brown eyes are dominant in the world population. And so, and we've simplified this, by the way. In all actuality, your eye color is not actually determined by one gene. It's actually a combination of genes. But for all intents and purposes, we say that, you know, in high school, that your eye color is one gene, because it just makes understanding this a little easier. But for example, your brown eyes are dominant. So if you carry the brown-eyed allele, you are automatically going to have brown eyes. Now, you have the brown-eyed allele from mom and a blue-eyed allele from dad, but because you have the brown-eyed allele from mom, you're automatically going to show brown eyes. Now, you can pass on the blue-eyed allele, which is why sometimes two brown-eyed individuals can have a blue-eyed baby, because that goes into the recessive trait there. The only way that you're going to have the recessive trait is you have to have both copies of the recessive allele, because it's normally covered up by the dominant trait if it's present. Blue eyes are recessive. That's why there's fewer blue eyes in the world population than there are brown eyes. Because the only way, like I said, you can show blue eyes is you have to get the blue-eyed trait from mom and dad. If you get only one copy of it, you'll have brown eyes. But you could pass on that blue-eyed allele, and then if your significant other passes on the blue-eyed allele, then your child could have blue eyes. But that's the only way you're going to have that's the only way you're going to show the recessive phenotype, is if you have both copies of it. So tomorrow what we'll do is we will get through some genetics problems. We won't get through all of them because there's a lot of different types of genetics problems that you can have, but we'll start with some of the basics, and we'll get into ratios and things like that. Like I said, I will go back over genotype, phenotype, dominant and recessive traits again, but overall, I just wanted to get those kind of in your brain there. So that kind of wraps up today. So hope you have a good day.